Yeah, thanks for being here. I was expecting a low turnout Thanksgiving week, you know, going home to see family and stuff. I'm leaving tomorrow morning. So anyway, I wanted to say first, thanks for having the, I appreciated Dr. Ballhorn to have the opportunity to give lectures. You know, I'm a full-time research scientist, so I don't teach in full-time, and it's really fun for me to do it, so I appreciate it. I also didn't mention last week that if any students are interested in doing any kind of project relating to this epigenetic inheritance, certainly feel free, I hope there are, feel free to contact me or Dr. Ballhorn. I've had, from previous years of giving these lectures, I've had, I don't know, 2 students who were interested in doing side projects, and they did. Of course, you know, you work in my lab, that's a tough experience. It's like slave labor, it really is. Because you know, it's 99% perspiration and 1% inspiration, and I get to do the 1% inspiration, and everybody else does the 99% perspiration part. It's not completely true, of course, but it's not true at all. But that's kind of like the reality. But I wanted to start today. After the lecture on Friday, a student came up and mentioned this issue of the giraffe. And that's that metaphor that Lamarck had, that the giraffe has a long neck because it's reaching for the higher leaves, and just, you know, so that's an adaptive, that's an acquired trait, reaching for the higher leaves. And of course that was dismissed by geneticists and evolutionists, thinkers, as not working, as not being accurate. And what this student mentioned was that there was a group of scientists who concluded that the long neck of giraffes was the result of a phenomenon calling sexual selection. In evolutionary thinking, Darwin was fascinated by this. There could be what is called runaway sexual selection. Or R.A. Fisher, that great scientist, also worked on this, and it was called Fisherian runaway. So a classic example of this is the peacock, where the male peacock has this huge feathers to attract the female, and biologically speaking, that's a maladaptive trait. It is not to the benefit of the male in some way to have this, to devote so much energy and tissue and so on to have these huge feathers. What happens is that the female are attracted to this display and choose the males with the fancier displays. And this pushes evolutionary selection rapidly. So you have sexual selection, rapid sexual choice selection for these gaudy display of tissues. So that's in evolutionary theory, that's natural selection versus sexual selection. Natural selection, you know, you have an environment, these birds are living in an environment, and over time, natural selection imposes selection pressure on them to adapt to their environment, whereas this is a maladaptive and happens very fast. Well, some thought that that is the case with the giraffe's neck, but it isn't. It's been kind of dismissed as an explanation, because both male and female giraffes have long necks, whereas in this Fisherian runaway or sexual selection, it's only the male or the female, one of the two sexes that has this, that develops this trait. So rapid Fisherian runaway sexual selection is not the explanation. But people are still trying to figure out, why do giraffes have long necks? Another possible explanation is enhanced vigilance, i.e. really long necks, their heads way up high, and they could see predators. Enhanced vigilance.
Don't they also use it as a offense against another male and dominance?
This is another example. There is some possibility of that as well. Here's my point, though. Doesn't matter what the adaptive trait is. These arguments don't prove or disprove whether epigenetics versus genetics plays a role in the mechanism of how this trait evolves. It could be epigenetic, there could be epigenetics involved, or genetics. Epigenetic variation or genetic variation. It doesn't disprove it. Just wanted to make that point. So whether or not the long neck of the giraffe is the result of runaway selection or enhanced vigilance or combat, whatever, it does not necessarily reflect a mechanism of that inheritance. And that's what we're talking about here today. Now, the next thing I wanted to do was, actually I was going to go back. I'll go back to that in a second, to something. Yes? Were you going to? Oh, no. Also, I forgot to include earlier on an important subject, which, I don't know, this is a plant biology class, and it's a very interesting subject, and there's a relevance to epigenetic inheritance in this subject. And that is the question of invasive species. I did have, there have been really interesting studies, and I think once upon a time I had a slide or two on the subject, but I didn't find it. But invasive species is an example of rapid evolution. So weedy species, there are species that have the ability to invade a new territory and do so rapidly. And if they're plants, they adapt to that new territory and do so within just a few years. And there was one really interesting study, which I didn't find in the time I spent working on this, where they found in California the species invading a hillside, and the morphology, the size of the seeds and the leaves altered in this population in just a few years, and was a heritable alteration that provided an adaptation to its new environment. So how does a population of an invasive plant adapt to a new hillside in a very short period of time? When I say short period of time, I'm talking about just a few generations. And there is a line of thought that epigenetic inheritance and adaptation plays an important role. Now, this scientist here, Christina Richards, is a very prominent person in this field. These are very good quality scientists, high quality scientists. This is a paper going back to 2012. There are more recent papers that she and others have authored on the subject, but you can see here by the title, Invasion of Diverse Habitats by few Japanese knotweed genotypes is correlated with epigenetic differentiation. And what they found was, summarized here, that I summarized from their abstract, that they looked at several of these knotweed species, different species, and they found that species to species variation, there wasn't much genetic variation within a species, it was more between species. So if there's not a lot of genetic variation, sequence variation within a species, how do a population of that particular species adapt rapidly to an environment? What they found is that there was little genetic variation among species, but in contrast, a great deal of epigenetic differentiation within each species, and evidence that some epigenetic loci, some of this epigenetic variation, may respond to local microhabitat conditions, i.e. epigenetic variation is providing the substrate, a term that we use, the substrate for selection for adaptation, rather than genetic variation. Now, if you read the literature, if you stay on top of this stuff, like I do, I'm fascinated by this stuff. I'm a plant biologist. You know, invasive species is an important subject. And all this is interrelated. So how a population adapts due to natural selection versus how a plant breeder achieves gain through artificial selection, it's really the same process ultimately. And so I just wanted to say that how this field is kind of still very contentious and very difficult, and a lot of controversy, is a review that I'm not showing here that they wrote just this year, just came out. It hasn't actually come out in print yet. That's not even the right word, in print, because they don't print it anymore. What do you say, come out in digital yet, or whatever? I saw just a draft of this paper, and they're still talking about, we have to be able to prove at the molecular level that this epigenetic change is directly causing this adaptation. Not just correlated with it, but causing it. So this field is very much still, ongoing, hot stuff today. Now, I wanted to go back because I thought that there was a student who was confused by a result that I had shown. And I just wanted to review it briefly before I get into today's stuff. This is my own work. And it's kind of small, but I think you could all see it. These are barley plants, Hordeum vulgare, it's one line of barley, which we grow with full nutrition, nitrogen, phosphorus, and potassium, or on reduced nutrition, reduced nitrogen, phosphorus, and potassium. And you can see a small plant, a healthy large plant versus a stressed small plant. We then harvest the seed from these two. Now, this is a cultivar of barley. These are genetically not identical, but this is a cultivar. There's no great amount of genetic variation between these two plants. The only difference between these two plants is the environment they grew in. That is the difference in nutrient supply during development. So then we harvest the seed from these two plants and then grow them in the next, this is just one type of experimental approach, grow that seed in the next generation side by side under a similar nutrient environment. And what we're looking for is differences in gene expression between these two plants growing under the same environment, the only difference being the environment their parents or grandparents were exposed to. And that's shown here. Now, this was the first example of one gene, but I then had this table, which is a complicated table, and I probably shouldn't have put this on a slide to show you guys. But let me just explain that what we're doing is we're extracting RNA from the roots and shoots of these plants, and then doing RNA-seq. So how many people here know what RNA-seq is? Anyone? Okay. RNA-seq sequencing is you extract the RNA from the tissue and you measure the transcription from every gene in the genome. And in barley there's approximately 25,000 different genes. So you're actually looking at the level of gene expression for every single gene in those tissues. And you could then look at the level of gene expression in plants whose parents were not exposed to stress versus plants whose parents were exposed to stress. You can look at the gene expression in those two types of plants, tissues, and you could actually, you get huge data sets, it's big data, but you could actually look in the data at each individual gene and say, is there a difference in the level of expression in this gene if the parent was stressed versus not stressed? And that's what I've done here. So this is root differential expression. The expression level here the expression of a given gene in a parent that's stressed versus the parent that's not stressed. It's actually log base 2 of the ratio. And then in the progeny of a parent, the progeny of a parent that was stressed versus the progeny of a parent that wasn't stressed. But in this progeny generation, they're grown under similar non-stressed, identical environment. And what we're seeing is for those genes where we see differential expression in the parents that are stressed, see like 1.3 log base 2, 3.7, 2.1. So the log base 2 of 2, so if there's a doubling of expression, you'll get a log base 2 of 1. So in this case, there's more than twice as much expression in this individual versus that individual. Way more, 3 times as much expression in this individual versus that. Now, if you see a differential expression in the parents due to stress, non-stress versus stress differential expression, then we ask, is there going to be inheritance of that differential expression in the progeny here? And what we find is that in roughly half the genes we looked at, the answer is yes. That is, if that plant's parent was stressed, the offspring shows an elevated expression of this gene, even though compared with the plant whose parent wasn't stressed. And so that's transgenerational differential expression. And we see this for about somewhere between 10% and 50%, depending on the experiment, of all genes. Of genes so what we're doing is, I mentioned to you in the last lecture that the field of study of abiotic stress response is huge. There's a lot of scientists out there looking at various kinds of abiotic and biotic stress, including Dr. Bellhorn, like for instance drought stress, or nutrient stress, or disease stress. It's a huge field, and they show, well, this organism, when it's being stressed, it's responding to that stress by expressing these different genes, increasing the level of expression or decreasing the level of expression of a bunch of genes in response to that stress during its life cycle. What we're saying is that those changes are then transmitted to their offspring. A significant fraction of those changes, not 0.1% or 0.01% of the genes, of the 25,000 genes, but 10%, 50%. And when people do studies of abiotic stress response, in my case, I'm very interested in phosphorus sensing and how a plant responds to reduced phosphorus supply. People who study this typically find, if you read all the literature, if you go on, get a PhD and spend months reading papers, or years reading papers, what people often find is, if you look at the differential expression across a genome in response to a given stress, you'll find something like 500 genes whose expression is altered in response to stress. Out of the 25,000 genes, there might be 500 whose expression is altered in response to stress. And this number, 500 or 600, is pretty generally true, whether it be drought stress or nutrient stress, various stresses. So there's something like 500 or so genes whose expression is altered in response to stress. And what we're finding is, intragenerationally, during the life of that plant, what we're finding is that of those, what we did was we took the 500 genes that people have identified that are differentially expressed in response to phosphate or nitrogen stress, and we asked, do we see transgenerational differential expression? Do we see that differential expression transmitted to the offspring? And of those genes, we see 10 to 50%. The answer is yes, we do. And now you guys are just students. You're not totally geeked out and haven't been studying, say, phosphate nutrition in plants for 30 years. So just to give you an example, when I was a postdoc 35 years ago, a graduate student in the lab that I was in finished his PhD and went on to get a faculty position and ended up identifying with others this particular gene here, PHG1-1. This gene, this is a phosphate transporter gene. And this is a big deal, this gene. A lot of labs, a lot of scientists around the world in yeast, studies with yeast, studies with plants, have been studying this phosphate transporter. So it's it's like a family member, you know? So I've known about this gene, and I have colleagues that have been working on this gene for 20, 30 years. And it's well known that this gene's expression, PHG1-1, and related genes, that gene shows differential expression in response to reduced phosphate supply. So the plant's growing, and it has a mechanism to sense how much phosphate supply it has. If the phosphate supply is low, it turns on the expression of this PHG1-1 gene so that it can take up more phosphate from the soil in the roots. So the roots will, it'll turn on that gene, crank up its expression. So there's more of this phosphate transporter so that it can transport more phosphate into the plant because it senses it needs more. This is how plants do things. So look, the log base 2 differential expression in the parent for this gene is 2.3. So that means it's got like 2 to 3-fold higher level of expression of this gene, making more of those phosphate transporters. And in the offspring, 0.9. Oh, I picked the bad case. So the answer to this question is that we don't see transgenerational differential expression. But in several of these other genes, we do see this differential expression. Actually, not the greatest example I picked to show you guys. But in other studies, that's actually near a cutoff. So the cutoff is 1, but it's 0.9. Didn't quite make it. Maybe with more replication, we'll show that. I can't wait to contact my friends who've worked on these genes for years and say, hey, you know what? There's an element to this biology that you guys have entirely missed, that you guys are not paying attention to. And that is that how organisms respond to some stress or change in environment is not an element of the selective advantage to that response is that it's transmitted to offspring. Because if you're a plant and you're under some kind of stress and you change your gene expression, doesn't it make common sense that if you could transmit to your offspring that change in gene expression, that would be a benefit to that offspring, right? Because now it's going to be better suited to live in an environment that it's living in. And that's really what we're talking about here. Did I lose all you guys on that? Kind of got it? Good. Let's move on to today. Now, today I'm going to talk about, I'm going to continue on with this stuff, and there's going to be some kind of modeling you know, theoretical stuff. And I hope that you enjoy it. We'll see. So today we're going to talk about epigenetics of multigenic traits that are conferred by selection for random epigenetic variation. If a plant senses it needs to increase the level of expression of a gene, if it senses it needs to do that and has a process to do that and can transmit that change in gene expression to offspring, that's not random. That's not a random process. There's a mechanism to do it. But now we're also going to introduce the concept of random epigenetic variation. So the substrate for evolution in the modern synthesis model, you know, the accepted model, is random genetic sequence variation. The substrate is in the modern is random genetic variation, sequence variation, and the rate of this variation, provides the substrate for evolution. The rate of this variation is one hit per base in the genome, one hit per base per cytosine, guanine, thymine, etc., per 1 × 10⁹ individuals. I don't know what that is. Is that a billion? That is a billion. So there's one hit per base in the genome, just randomly, regardless of environment, in 1 in a billion individuals. The substrate for evolution in new synthesis model that includes epigenetics, in random epigenetic variation, in contrast, and that is going from a cytosine to a 5-methyl cytosine, methylation of a cytosine, any cytosine. That's 1 out of 4 bases in the genome, is one hit in 10⁴ individuals, 1 in 1,000 versus 1 in a billion. So if here's a gene and here's a cytosine, the rate of mutation of that cytosine to a thymine, genetic change, 1 in a billion. That same cytosine, the rate of methylation of that cytosine to a 5-methyl cytosine, 1 in 1,000. Much higher rate. Orders of magnitude higher. And we're going to talk about what that means and how we might study it. This is very cutting edge stuff. Let's look at, and I like to give examples. That's what all these papers that I show you on these slides are examples of this stuff. Here's an example that's relevant to climate change. Response to multi-generational selection under elevated CO2. Climate change, elevated CO2, plants have to live in it. People are very interested in this subject, to say the least. The response in multi-generational selection under elevated CO2 in 2 temperature regimes suggests enhanced carbon assimilation and increased reproductive output in brassica. Again, a lot of these studies are with brassica for some reason. I don't know. So if you look over here, starting population, this subset that are treated, that are exposed to elevated CO2 in going from the 1st generation to the 4th generation, that's just 4 generations. Then they find that there's the seed yield is pretty significantly increased after just 4 generations if the population was exposed to elevated CO2. So in 4 generations is not a lot of time. In just 4 generations, there's been a heritable change in that population. Those plants are being more reproductive. They're making more seed and heavier seeds in the high CO2 versus plants that were not exposed to CO2 for those 4 generations. They have all the proper controls in it. So how could that be? Yield increase observed after 4 generations of brassica when grown under high CO2. Remember, these are controls. So they have the 4 generations grown under high CO2, and then see how much they yield under high CO2, and then take the control, which wasn't exposed to high CO2, and grow it under high CO2. Yet you see this big difference in the plants whose parents were exposed to high CO2. So they're adapting to their new environment, which is high CO2. Is this gain due to selection? What do I say? First question. Is this gain due to selection? Is this gain? Has there been any selection? Did someone, like these scientists, grow 40 plants and pick the biggest ones? Do a selection? The answer is no. No selection was involved here. They grew the population, harvested the seeds. there still could be a form of selection, and that is plants that produce more seed per plant might contribute more seed if you just harvest them all. Those happier plants provide more seed to the total amount of seed. So that could happen, but there was no actual selection in this experiment. I looked at it carefully. Do these plants sense elevated CO2 and then adapt via induction of favorable programming? That is, in this question of induced versus random. And I think that's what's going on here. Point is, I just want to start thinking about this because that's what I want to talk about. That's what I want to talk about today. If there's non-random change going on that's heritable, the environment influences gene expression and that's passed on to offspring. If there's that going on, but there's also random variation going on, high rate of random epigenetic variation going on, how do you distinguish the two? How do you quantitate how much the random epigenetic variation is contributing to evolution versus the non-random variation? Again, guys, when I talk about this stuff, I'm a plant biologist and I like evolutionary theory and I'm a geneticist. Got to always remember, all the stuff is highly debatable and controversial. Just want to make that clear. Don't think that anything I talk about means that if you go out and do a random survey of scientists in this field, they'll say, oh yeah, that's how it works. This stuff is all very current and somewhat controversial. So, also, is this a single gene trait? Is this difference here due to a single gene? Not likely. It's a quantitative trait, no question about it. So, now, let's just take a moment. This is important to understand really what I'd like to talk about today. Simplified model of Lamarckian versus neo-Darwinian evolution. Increasing adaptation in response to selection pressure in a population via either Lamarckian, or I wrote this several years ago, I might not call it that today, Lamarckian versus neo-Darwinian evolution. So look, this way of thinking about this actually was first described in a book by two scientists, Jablonka and Lamb, in 1995. This was an important book. They theorized this stuff in 1995. That's over 20 years ago. And this was their figure. Well, actually, it's a copy of their figure that I've modified to describe what we're thinking about on this stuff. See this box here and these circles? Each circle is a plant. It could be an animal too, but let's just consider this as plants. Each circle is an individual plant, and this box represents, say, the side of a hill, a hillside. And you got this population growing on this hillside. And this, going in this direction, are generations. First generation, or zero, the beginning generation, 1, 2, 3, 4, like this. So this is generations of this population growing in some hillside. And let's first look at neo-Darwinian evolution, the classical, the modern synthesis, the way people think things work. If this population of individuals is growing on a hillside and there's some selective pressure, like for instance, increasing temperature, decreasing water, any other form of biotic or abiotic stress, I'm going to take a moment to say to you, what do I mean by stress? All organisms, all plants in a field, in a farmer's field or out in the wild, and all animals live under stress, are living in an environment which represents a challenge. And all organisms are trying to deal with that challenge by being reproductively successful for the next generation. So stress is just reality for all organisms. So it's not some special thing, like, whoops, oh good, like climate change. Climate change is not some unique special thing. It happens. The whole thing about the debate about evolution is it's really like nonsense. Evolution is going on all the time and has been. People argue about whether evolution is a theory or a fact. Science has gone so far beyond that. Now we're trying to understand how it works. But the fact that we don't completely understand how it works doesn't mean it's not a fact. It's still a fact. The metaphor I like to use is gravity. So people say evolution. Well, they don't know exactly how it works. It's all theory. It's all it's just like a belief. Well, what about gravity? We don't know how gravity. Actually, we getting to the point where we do know how gravity, thanks to Einstein, getting to the point where we might start to understand how gravity works, but not really. So we don't know how gravity works, but is it a fact? So what I like to do is challenge any skeptic of a tall building and say to me that we don't know how gravities work, but let's do an experiment. You jump, and we'll see, whether it works or not. So here's this population, and there's some stress on that population, and there's one individual that has a favorable genetic variant that makes that one individual more successful reproductively in that environment. And so over generations, that one individual contributes more progeny to the population, becomes that valuable variant becomes more, this is quantitative genetics, population genetics, that allele becomes more common in the population because it enhances the individuals that have that allele, it enhances their reproductive success. So that over time, that genetic change becomes widespread in the population. That's classical evolution. That's the modern synthesis. So favorable allele provides reproductive success and becomes more and more widespread in the population, and over time, that's how populations evolve. But I show this through 4 generations, starting from a favorable allele to being that alleles throughout the population. That's scrunched down in time. This could take way more than 4 generations for an allele, a beneficial allele to spread in the population. Hundreds, 10 generations, 50 generations, 100 generations. I scrunched it down in time. Or at least Jabonka and Lamb scrunched it down in time. Now in Lamarckian evolution, there's a difference. What we're talking about, I shouldn't have called this, I should change this and use a different term, not use a name, Lamarck. But in this new model, this population is under some stress. And in this new model, each individual in the population can sense that stress and change its gene expression. Not one rare mutation, but each individual. When I talk about plants sensing phosphate, say there's 1,000 corn plants. I'm not saying that one of those 1,000 corn plants senses they need more phosphate and increases the level of expression of phosphate transporters. All of the corn plants, each individual corn plant, senses it needs more phosphate and changes its gene expression. And each individual can transmit that change to its offspring. So each individual in the population senses this stress and increases gene expression, adaptation, and that increases over time. The whole population, see, the difference here is the whole population is evolving, not just one individual responding. The whole population is moving forward because each individual can sense its need and respond to it. So the whole population moves forward. This is very different. These 2 models of how things work are very different from each other. If you model this mathematically, you'd have a different equation. It would be a different equation that would describe this sort of pattern versus this sort of pattern.
I have a question.
Yes, please.
What were the original labels on that model, if that was from 1995?
I don't remember. Yes, this is a faithful reproduction of what they showed, but I don't know if there was any specific label on it. What the stress was, what the environment was. I don't remember.
Getting at something similar that you are describing?
It was similar, yes. This is basically what they're trying to describe. Yes?
So which model should we actually follow? Because when we get into biology, we are thought about neo-Darwinian evolution. That's what we are thought.
Right.
But
I don't think that I could tell you which you should believe in or think is the right model. As a scientist, I'm just showing you what people are doing and what people are observing, and then what the conclusion is. I think the answer actually is both are happening. So I don't think it's Lamarck versus Darwin. I think it's both things are going on. But the struggle is to understand how. So we made a lot of progress in understanding how Darwinian evolution, the modern synthesis, the standard model, we made a lot of progress in understanding how that works. Huge amount of progress. We haven't we're still working on how this might work. And so there's still a lot of work to be done. We're working on this literally today, you know, not today, I'm talking with you guys today, but actually today. So as a student, I wouldn't think either is, well, there's a lot of support for this, and this is an up-and-coming new kind of view. If you recall, I don't know if you would, because there's a lot of information that I'm throwing at you guys, but one answer that a doctor might give to your question was that doctor in England who, when asked about that same question, is this happening or is this happening? And he says, I'm a doctor. And what he meant by that was that if these are humans, and these are the parents and the children, and some disease or some exposure of the parent alters the phenotype of the children, he doesn't care what the big theories are. He has to understand why, how, what is happening in these individuals. How could he treat them? If we know that if your grandmother, if you're a woman, and your grandmother smoked when she was young, you on average are going to have a higher incidence of some disease. That's something that a physician would like to know about, you see. What the mechanism of that disease inheritance is where people are still working on it. There's actually been some progress, by the way. I can't show all the stuff. I mean, I'm fascinated by this stuff. There's an example, a well-known, one of the first examples of an actual molecular mechanism for this in humans was that they found that individuals whose, and this is quite relevant in our world, when you look around what's going on in the world, individuals whose parents were in the Holocaust, grandchildren of Holocaust survivors had certain genes turned on due to the fact that their grandparent was in the Holocaust. They have identified the gene and the change in gene expression in the Holocaust survivor. Because there's in the Jewish world, there's a thing, well-known thing, a Holocaust survivor syndrome. I actually had a friend whose parents were in the Holocaust, and he had mental illness due to that. So it's happening. There's inheritance of traits that is not Darwinian or Mendelian, I should say Mendelian in nature. And the question is understanding. That's a long, sorry for such a long response. Yes.
So it seems like a lot of the studies that you're talking about shows like 6 nice things that happen from stress responses, epigenetic. And what you're describing in humans are detrimental.
Very good question.
Detrimental effects you've noticed in plants.
Right, there are. I didn't have enough time. I sort of brushed by it very fast last Monday or whatever it was, last Wednesday. So there's adaptive change and maladaptive, bad stuff. Both happen. So the example that I gave last week was studies of drought response in plants. And so, and this is, thank you for this is a good question, and it's important to understand this question. In the field of plant biology, in agriculture, understanding drought, adaptation to drought is a huge issue. Can you imagine? You know, throughout the world, because there's a lot more drought today than there was 20 years ago, or 50 years ago. So understanding how, so figuring out the genetics of drought tolerance is a huge field of research. Corporations, Monsanto, DuPont, are all working on figuring out a genetic approach to imparting drought tolerance to corn and soybeans, etc. Huge field. And the model organism for a lot of the research, the model was this plant called Arabidopsis, a small plant you grow in the greenhouse. And scientists were exposing Arabidopsis to severe drought and identifying the genes whose expression was altered in response to that severe drought. And the question there was like 400 genes. I actually met a person working at DuPont on this. They had like 400 genes that looked like they were important in drought response. But they couldn't transfer that scientific finding to corn and soybeans. It didn't work. They identified genes in Arabidopsis that were drought response, that were responsive in severe drought, but then when they tried to manipulate those genes in corn, it didn't work. They didn't get high-yielding corn plants. The reason for this is that responses to severe drought are not adaptive if the response you're looking for is to moderate drought. Give you an example. Stomata, this is the one I mentioned, those are the holes in leaves. In severe drought response, the stomata completely close. And if you figure out how that works, and you could induce that in a corn plant, great, right? It will survive under severe drought. But if the stomata are closed, the plants are not transpiring, and if anybody studies plant biology, plants need to transpire to move water through to grow. So that adaptation to severe drought was maladaptive under moderate drought. Do you understand what I'm saying? And that is key. What people need to do if they want to figure out drought response is study the adaptation in the environment that's practical, that's a real world environment, the moderate drought. Because plants are not out there with no water, they're out there with reduced water, you see. So there's no question that any of these changes could be adaptive or maladaptive. And in fact, I talk about adaptation because that's what people talk about. But ultimately, that's a tough word. Because what is adaptation? Adaptation is a very relative thing. It's adaptation to a given environment. But what if you have a different environment? There was a story that just came out this weekend, about the sockeye, excuse me, was it the sockeye salmon? I think it was sockeye salmon in a nursery that they released all the baby fish. They grew them in a nursery here in Oregon. Was that in Idaho? I forget, which I think it was Oregon. They released all these fish to go out to sea, and they all died. Why? The water conditions were too different. The environment they were growing these fish in was too different. So it's all about the environment we're actually in. It's all relative, in other words, as to what is adaptive and maladaptive. Did I answer your question at all? Yeah. Okay, we'll move on. So, a couple of things more. Lamarckian versus Neo-Darwinian. Same two figures. Let's ask a few questions. What are the implications of this pattern, where all the individuals in a population are slowly adapting, versus this pattern, the Darwinian or modern synthesis model, where it's selection for rare allelic variants? First fact to take into account when we think about these things, effective population size. In Darwinian evolution, in Neo-Darwinian evolution, in modern synthesis, what do we think about? Big populations, right? I mean, you need a pretty big population to have an individual that has the rare beneficial mutation. Genetic change, 1 in a billion individuals. That's for a single base change. You need a pretty big population for this phenomenon to be happening. How likely is it if you had a population of 45 individuals and you change the environment on them, like plants on a hillside, that there's going to be some rare beneficial mutation? It's not likely. So there's a different, how about this model? What is the effective population in this case? The effective population, so this is what geneticists think about these things. So if you want to find some trait, you got to think, how big a population do I have to grow to find a plant that has that trait in it? It's a practical consideration for geneticists. How big a population do you need for Lamarckian evolution? I always play this game. What do you guys vote on? What do you think is the effective population size for Lamarckian evolution? I'll give you the answer because we're always going to be short of time. 1. If the mechanism is that each individual can sense its environment and adapt, you just need an individual, one individual. I once got in trouble on this question because the USDA, the US Department of Agriculture, maintains collections of different crops like corn, germplasm collections, because in the thousands and thousands of lines of corn and barley and wheat that have been grown over the years, there's going to have all the different wild barleys, wild corn, wild wheats, cultivated wheats. In that collection, there's going to be all good genes. And I was actually interviewed to oversee one of these collections. And I told them in the interview, I'm like this, you don't need this collection. We don't even need this collection. Because you just take a few corn plants, a few barley plants, we'll be able to breed, just starting with just a few plants, we'll be able to get what you want. You don't need tens of thousands of lines of corn and barley to find our favorite gene. All we need is a few. They didn't like that. because the whole reason to be. Now, evolutionary time scale. How much time does this modern synthesis need to occur versus this Lamarckian model? In our classical notion of evolution, we're talking hundreds, thousands of years. Not short-term. All these studies showing short-term evolution. How does that short-term evolution work, is the question. You don't have thousands of years for this population to adapt to its environment. It adapts in 1 generation or 2 generations. How does that work? So, key paper. These guys have done a lot of important work. Lead scientist here, Frank Johannes. Big group, big work. Rate, spectrum, and evolutionary dynamics of spontaneous epimutations. Here's the bottom line. You could skip everything and go down here. The above rate for individual bases, genes contain, the rate that I mentioned to you, 1 in 1,000 versus 1 in a billion, that's for single bases. But any given gene has 5,000 bases. So what's the rate at a gene level? If we divide the above by 10 to the 3, like 1,000 or 5,000, 5 times 10 to the 3, the rate of forward genetic mutation is about 1 in 10 to the 6, or 1 in a million. So the random rate of mutation on a per gene basis is about, there's a mutation in any given gene in a million individuals. The rate of epi-mutation is somewhere between 1 in 10 to the 1, that is 1 in 10 individuals, or 1 in 10 to the 2, somewhere in there, 10 to 100. In every 10 to 100 corn plants in a field, there's a change from a cytosine to a 5-methylcytosine in a gene, in every gene. Epimutation, epigenetic variation is going on all the time. It's going on here. If you guys, I hate to say this, I shouldn't say that. Okay, I will. If you guys smoke pot, there's a chance your grandchildren are going to have some weird thing. Keep laughing. If you guys eat Twinkies too much, your children and grandchildren can show effects, even if they don't eat Twinkies. You see, that's the point. Even if they don't smoke pot. It doesn't matter what their environment is, it's your environment. You're adapting, you're responding, and you can transmit those responses to your offspring. Not can, will. Weird stuff. I'm sorry. I think about this a lot, I really do. Now let's introduce this concept of random epialleles. So this is that same figure again. Is it already 20 after? Holy mackerel. You see, now I've slowed down. And I'm not good. But let's look at this. I'll wrap it up fast. If you have, this is the induced epialleles, the first example, where it's a non-random process. And this is the random alleles. And this is introducing the concept of random epialleles. So what happens is, if you're making gain due to selection just based on random epialleles, the rate is still going to be far greater than genetic alleles. You'll get a fixation in the population of some trait much faster due to random epialleles because the rate of epiallelic change is so much greater than genetic change. And it's happening all the time. So I'm going to skip over this because it's going to run out of time. Skip over that, and get to this. Energy use efficiency is characterized by epigenetic component that can be directed through artificial selection to increase yield. What these guys did, and I'll just summarize it for you, and we'll skip over all the hardcore data because we're going to run out of time. But I could explain it to you real quick if you like. What these guys did is they took a population of canola. It's also brassica. It's another brassica. Canola. Canola oil, right? You guys know that. See, I tell you so many stuff. When they were trying to grow canola as a crop in Montana, they were looking for a name. See, canola is Canadian oil. That's how it got its name, canola. Canadian oil, canola. So now they want to have a market to grow canola in Montana. And I was in Bozeman, Montana, and I said, how about Bozola? That didn't catch on. What they did is they grew this canola, and they had only like 200 plants, and they sampled leaves from each plant, and they tested respiration rate in that leaf. And they took the 5 plants from 200 that had high respiration rate, and the 5 plants that had low respiration rate, and bulked the seed from those. This is called recurrent selection. They identified in 200 plants those 5 that had the lowest respiration rate and those 5 that had the highest respiration rate, and harvested the seed from those plants, and then had 2 populations. Progeny of plants that had high respiration rate versus progeny of plants that had low respiration rate. Now, no selection pressure, no stress, no low phosphate, high phosphate, heat, nothing like that. Just grow the population, measure the individuals for respiration rate, bulk the seed, and then grow it in the next generation. And they found that after just 3 to 5 generations of this recurrent selection process, again, no selection pressure whatsoever, just selecting for those individuals that have high respiration rate and those that had low. After just 3 to 5 generations of this sort of recurrent selection, they were able to show improved energy use efficiency, higher yield in the progeny whose parents had low respiration rate. What are they selecting for? There's no, in 200, another point I want to make about this study that's important. This population they started with is so-called doubled haploid population. What this is they took an individual and what it is, it's a clone. That's the easiest way to express it quickly. This population is a clone of genetically identical individuals. 200 genetically identical canola plants. And they just identified 5 that had a high respiration rate and 5 that, well, why, if they're genetically identical, why is their respiration rate vary? Why is there variation in their respiration rate? Because there's variation in populations that's not genetic in nature. It's epigenetic in nature. And you could select for that epigenetic variation, just like you could select for genetic variation. But you could never achieve this gain in 5 generations, 3 generations. You can never find increased yield in 5 generations or 3 generations if that required, in a population that's genetically identical, that's a clone, basically, if you were selecting for some sort of genetic variation. It's not going to be there. The numbers don't add up. So the conclusion is, and they demonstrated this, that what they were selecting for was epigenetic variation. And you could achieve gain in just a few generations. This is a critical study. And it could explain a lot. I'm going to skip all this. That explains it in detail. And just go on to this. So how can we explain the ability of these guys in the Netherlands? How could they achieve gain due to selection, artificial selection, in small population, remember population size? In a small population in a short period of time. Selection for epigenetic variation, which they did document. So, you know, I'm going to run out of time. I don't know how did the time go by so fast? Did I not talk so fast? But let me tell you about, because somebody asked about this, and I must show you this. I'm going to skip all that and get to this. RNA-mediated genes. Somebody was asking me about mechanisms. And I'll explain this to you real quick. Again, I only need a minute if I explain it in words. Small RNAs. There's, I don't know how much molecular biology you guys have had, but you know, the basic model of cellular biology is you got a DNA, it's transcribed into an RNA, which is then translated into a protein. Small RNAs are short RNAs of 24 mers, 24 nucleotides in length, as an example. There's a variety, there's a spectrum of these things, but just say 24. And these 24 mers, these small RNAs, regulate the expression of genes. And here's the interesting thing. They are translocated, they can move from cell to cell. They've been found that they get packed into sperm. Guys, this is a male form of inheritance through the male. And, you know, pollen is sperm. They get packed into pollen. So now, here's the kind of way you're looking at it. Plants are exposed to some stress. There's gene expression changes. which are those gene expression changes are mediated by these small RNAs that turn on and off genes. However, they can be translocated and packed into pollen and then transmitted to the next generation. So the pollen fertilizes the egg and the plant grows, and it's got those small RNAs in it that are then transmitting that change that they were designed for to induce. And this is actually happening. The small mystery males at corn pollen and mice. I love this because it's the same in both organisms. Sperm. Lifestyle in the sperm. Studies coming out. The extent to which useful adaptations can be inherited epigenetically may depend on small RNAs packed into sperm. It's a mechanism. And lo and behold, you see, I've been talking and working on this for years and trying to get grants to work on it. Meanwhile, I haven't gotten the grants. My work is not progressing that fast, but other people are getting grants. And, oh, I'm going to leave you alone in just a minute. Europeans, I once had this finding that I told to a person leading funding for this research in Idaho, this epigenetics inheritance, and that person said to me, oh, we're not interested in that, but the Europeans are, quote, unquote. And yes, the Europeans are very interested in this stuff. And again, the same group showed that small RNAs reflect grand-parental environments in the apomictic dandelion. So they document that, in fact, these small RNAs that are packed into sperm, into pollen, the apomictic dandelion doesn't make pollen. So I'm not exactly sure that's a perfect proof of that. It's a clonal propagation system. But they show that the small RNAs are playing a role in transmitting these changes in gene expression. So I'm going to have to skip over all of this to say the one last thing. And then I'm going to let you guys go, and I still got 2 minutes. Here's what's really important about this field. How can epigenetic inheritance play a role in genetic inheritance? Is there some way that epigenetic changes in gene expression can be permanently captured in genetic information? And I call it it hasn't been proven. It's a total hypothesis. The epigenetic two-step. You go from cytosine to 5-methylcytosine, and then that 5-methylcytosine gets converted to a thymine. Cytosine, 5-methylcytosine, thymine. You know, here's the bottom line, the conclusion. This epigenetic inheritance is happening. We don't know exactly all the details, but if there's a new science that impacts inheritance, and it's really revolutionary, it's really kind of revolutionary and new, what that anticipates is that there's going to be new phenomena and mechanisms that we don't currently understand that are yet to be discovered, and they will be discovered probably relatively soon. New things that we had no clue about, like paramutation, like small RNAs. Perhaps this epigenetic two-step where you have an epigenetic change going from cytosine to 5-methylcytosine, then you capture that permanently as a genetic change, conversion of 5-methylcytosine to thymine. So I'll leave you with the last thought. The most common genetic change in genomes is in fact this one, cytosine to thymine. That's the most common genetic change. Maybe all along this epigenetic two-step's been going on, and that's really what it all reflects. So I hope I didn't give you guys too much of a headache. If anybody's interested in this kind of thing, this stuff, wants to do a side project, some kind of project, contact Daniel or me. No guarantee we can do it, but if you're interested, let us know. And I hope you enjoyed it. Thank you so much for being patient with me, and have a nice Thanksgiving.